I'm not really a drinker, not a smoker. It was devastating.
Head and neck cancer is one of the most common cancers worldwide. Half the patients with oral cancer, the most common type of head and neck cancer, die within 5 years. While smoking and drinking alcohol increases people's risk of oral cancer, doctors are seeing more and more patients who don't do either. And black patients have worse outcomes than others because their cancer is caught later. We have discovered a gene that might be to blame, and slowing down the cancer forming processes driven by this gene may lead to new ways to treat head and neck cancer.
If we're able to target it, then we can arrest head and neck cancer and before it progresses to more advanced stages, or it may actually replace some of the treatment, which some of it is toxic. That would be very beneficial to our patients.
This gene is called PDCD10. Our early research showed that this gene increases the growth of the cancer and it stops treatment from working well, which is obviously bad news for our patients.
We did some analysis comparing patients, and that study showed that patients with high PDCD10 expression have worse survival than patients with low PDCD10 expression.
We are going to learn more about this problem gene. Our lab will grow cells in a way that mimic human cancer in a dish. Once we understand exactly how this gene works, we can create treatments that actually stop cancer by restraining this gene.
And it was a long process too, even after the surgery. I mean, for 3 or 4 months, my speech was probably at the 85 or 90% range. If we can evaluate a protein in somebody's mouth, it will save people from the devastating effects of a lot of the treatments.
Chemotherapy kills both bad tumor cells and good healthy cells. That's why a lot of chemo patients lose their hair and have other side effects. This project will allow us to create new treatments that target the genes that only play a role in the cancer cell. This way, patients could get better with less toxic treatments.